Hello, my name is Jordan and today I'm going to be talking about the blood-brain barrier. So the blood-brain barrier is super important to understand within neuroscience and pharmacology because it really affects what kind of treatments we can use to treat brain disorders. The blood-brain barrier is made up of three types of cells that form this really tight junction. It consists of endothelial cells, pericytes, and astrocytes. And this tight junction kind of serves as a protective barrier between what molecules and toxins or hormones, anything like that within the bloodstream. It kind of limits what molecules can pass over into the brain or into the neural tissue and surrounding brain cells. This kind of acts as a protective barrier to make sure no toxins or bloodborne pathogens can enter and harm the surrounding tissue. So because this barrier is so selective, only a certain type of molecule can pass through, and usually these are very small lipid-soluble molecules. So things like hormones or steroids such as testosterone, estrogen, or things like caffeine and nicotine are easily passable through this membrane. On the other hand, molecules that are large or polar or non-lipid soluble have a really hard time passing through this tight membrane. This would include things like proteins, possibly pathogens and viruses, or things such as water-soluble drugs like penicillin. Because this barrier is so selective, pharmacological treatments can sometimes be difficult because you might not be sure that your drug can pass this blood-brain barrier. So the main focus here would be to understand that a lipid-soluble drug can pass through and maybe focus on treatments such as that, or focus on ways that can alternatively affect the permeability of the membrane to allow for your drug or treatment to pass through. Ultimately, it's super important to understand the blood-brain barrier to understand how the brain keeps homeostasis and just keeps itself safe from any toxins or pathogens that may be included in the blood.